fruits and vegetables, have you ever wondered that how many chromosomes are present in them? How much DNA might be present in them, or is it possible to isolate the DNA easily? So today we are going to perform an experiment where we will going to isolate the DNA from very common fruits and vegetables. So let's get started. As we are using plant tissue, so we are using these fruits because it is better to take specimen from those fruits which have high number of chromosomes. Things required for the experiment: glass rod, a grater, 2 test tubes, a funnel, a large beaker, 2 small beakers, a mortar and pestle, a dropper, a dish soap detergent. This detergent acts as a source of lipase. We just need to dilute it a little bit so that we can add a little amount of water.
Now we will need the papaya. Why we need the papaya? Because the papaya contains the protein digestive enzyme called papain. Now we will cut the papaya into the fine pieces and grate with the help of grater so that we can extract the juice from it.
So today we will be taking banana as a source of DNA. So firstly we have to peel the banana and we will take it in the water first. Now we will making a paste of this. So for this we have to use some distilled water, not much but about 10 ml to 20 ml, and a pinch of non-iodized salt. And when we make this into a paste, we hope that while grinding it, converting into the paste, the cell wall of plant cell will break. Usually in biotechnology, we use cellulase to.
So we are trying to reach the DNA which is present inside the nucleus. So after removing the cell wall, the next membrane that we have to remove is cell membrane and nuclear membrane. So as we all know that cell membrane and the nuclear membrane consist of large amount of lipid on which proteins are floating.
So we will use the lipid digestive enzyme to break down the cell membrane and nuclear membrane. So what will we do? We stain the banana extract. Add soap solution for digesting the cell membrane or nuclear membrane. So we are using this is a source of lipase to break down the cell membrane. I am stirring that so that this easily dissolves.
Now we will be using protein digesting enzyme because we have cell membrane or nuclear membrane, but we have to reach the DNA because DNA is a form of chromosome, and we know chromosome contains DNA 45% RNA, 10% and protein 45%.
So even we do not have removed RNA, we have to remove protein, so we get DNA. So here protein digestive enzymes, we use papaya extract prime. Now we are stirring this.
Now we will need a test tube. We will take a small amount of solution in the test tube, and lastly we need chilled ethanol to create a density gradient. So that the DNA float out and get accumulated in the content and chilled ethanol. The DNA is accumulated in the junction between the chilled ethanol and solution. This white cluster part is DNA.